All right. So in this second lecture of the first week, I'm going to now start from the beginning of the class and give an overview of molecular biology and genetics and describe the data that we will be dealing with this semester, targeted towards computer engineers. So, as a computer engineering student, you may have forgotten your biology knowledge in high school, but that's okay. This introduction to molecular biology and genetics should be sufficient for you to follow the rest of the material this semester. All right. And about last Monday's lecture, if you have any questions, please let me know. There was the second hour of that lecture was used to give you a glimpse of what you will see, what kind of theoretical analysis or how we're going to analyze the algorithms and describe the algorithms that we will be talking out throughout the semester. And if you don't have any problem in following the material on Monday's second lecture, you're welcome to write your name in this form, Google form, which is Introduction to Bioinformatics Additional Enrollment List. If you're not a fourth-year student, if you're not able to get the course in a regular manner with the token assignment system, please feel free. And if you want to take the course with credit officially, please feel free to write your name, surname, email, which department you're from, class, whether you're a third year molecular biology and genetics student or a master's student from Informatics Institute, Bioinformatics program, and also write your student ID in 7 digits. So the link to the form, the capacity of the course is, I don't know, it will probably be close to 100. There will be I will not limit the number of students who take the course. Okay, so it will be unlimited. So let me share the form with you again. So this is the short link for the form. So if you want to take the course, you'll be able to take the course. To everyone. All right, so this is the link to the form. And also the course videos are going to be in this playlist. I haven't shared this playlist with you in our departmental news group because there are lots of students from other departments which do not have access to our departmental bulletin board system, the news groups. That's why I'm going to mainly use Oticlass for discussions or announcements related to this course. But since nobody is in Oticlass as of now, so these two links that I just shared in this class is only available when you're joining these synchronous sessions in class. All right. So, and which is also available on my personal homepage, the first meeting information and links to the synchronous classes are available in my personal homepage under courses this semester. So you should be able to join these sessions. All right. Anyway, so I will be using the same playlist to upload videos that I record throughout the semester. So make sure you somehow bookmark this link. They will be unlisted, so when you do a search for Introduction to Bioinformatics on YouTube, you will not be able to get this playlist. Okay, since the videos in this playlist are unlisted, it may not be returned as a query result when you do a search on YouTube. And I also have some previously recorded videos on YouTube on Introduction to Bioinformatics, which are really old, like maybe 10 years ago now. But the lecture material is pretty the same. You can also watch those videos if you want. Anything you want to ask about administrative stuff related to the course? Do you have any questions? Anything unclear? Oh, I just came up with something. This form will be available to fill in until tomorrow, Friday, 5 p.m. So last time to enter your name enter your name in the form is Friday, which is the 19th of March, Friday, March 19, 5 p.m. Okay, so I also wrote that in chat box. And after that, I'm going to just submit this list to our department secretary, and she's going to enter your student IDs as additional course acceptance criteria. So you will not be automatically added to the course, okay? So having written your name here does not make you this course appear in your registration system. You still need to do it manually. This list is used to enter additional criteria in the system so that you can add the course, okay? And if you had written your name in this course and changed your mind on Monday and you decide you don't want to add the course, that's fine. All right. You don't need to, there's no need to erase yourself from this list if you don't want to take the course. If you don't add the course on Monday or in the add drops, that's okay. But if you want to add the course in the add drops, you have to be on this list by tomorrow, 5 p.m. If not, it means you have lost the chance. You should have attended the first weeks of lectures. You should have shown an interest in taking the course by attending the first week's lectures. Otherwise, I mean, if I get emails on Saturday or Sunday or next week during the add drops saying that you will need to take the course, it's too late. All right, that's the last time to enter your names. Oh yeah, it's not for computer engineering students. Computer engineering students, they do not, these are only for irregular students who cannot take the course by using the token assignment system. If you are in the token assignment list, that list is also going to be automatically added. So yeah, to the system as criteria. Jang, as four-year students, you should use tokens, but I don't think the token is going to be the token limit is going to be too high for this course. So, I mean, I got some questions about, since I basically give unlimited capacity to all the students, it should be a little bit like unfair to fourth-year students who wants to spend some token on this. Maybe I should talk with Emre Hoca about this, but I mean, yeah, that's and also exchange students. The reading materials on the course website. I forgot to mention that. I will resend this link when you're registered in OT class. There's a username and password which is not auto username or password. So the reading material, some of them are copyrighted material. The username to access this is CENG465 is the username, and the password is 5710465 underscore. Okay, so that's the password. So if you write this username and password to access the reading material, you should be able to access it like that. And the lecture slides should all be publicly available. But yeah, the reading part, as I said, since it contains some copyrighted material, you should use the password and the username that I just pasted on the chat box to access the reading material on the course website. And yeah, I was talking about fourth-year students in our department. Yeah, they should get the course within the token assignment system. All right. So this, since the other students from other departments, since this is an interdisciplinary course and it's a must course in bioinformatics department, for example, in the MS program, so they need to take the course. So there are some students that should take the course outside the token system. They are not part of our department, so I should give extra capacity for them. And normally, fourth-year students in our department should spend tokens to get the course. But in case you cannot get, I will give additional. I will increase the capacity for students who cannot take it from our fourth year. If you have other, I mean, if for example, you're a fourth year student and missed the token system, you're welcome to write your name. So any irregular students who wants to take the course and cannot take the course via the regular token assignment system are welcome to write their names and surnames on this, and student IDs in this list. This form here, the Google form here, is on this chat box. Let me repeat it again. This is the link to the form, which is only available via the synchronous lectures, and this is the link to the YouTube list. Okay, so this is the link to the form to add your name. It should be available. Let me ask you a question. If you join the synchronous class, this meeting, Cisco WebEx meeting, like 5-10 minutes late, do you see the earlier chat transcript, or just like, for example, in WhatsApp, for example, if you're just added as a new member, you don't see the previous post by other members. So when you join late, do you see these previous chat transcript that we had written, we have here? Or do you just see the chat transcript starting after you join it? Oh, you don't see the previous chat transcript. Okay, so let me copy the form link again for those students who have come in late. Okay, you see after you join. Or maybe what I will do is, so this is the, all right, so it's just like what's up. So these are the links for the form and the YouTube playlist again, okay? And I will also post these two links in the video description today. Okay. Now, do you have any questions about enrollment or other administrative things? If not, what I'm going to do is, as I said, I'm going to start with an introduction to targeted towards computer engineering students, molecular biology and genetics introduction, not the syllabus, the lecture slides. All right. All right, so I'm going to skip the syllabus related material we had last time. Yeah, we are right here. So what is bioinformatics? So some of these slides are taken from Mark Gerstein's bioinformatics course slides. So I gathered different material from different available slides or my previous slides that I prepared. So it's really a collection of different material from different places. So bioinformatics, what is bioinformatics? Many people call different things bioinformatics. Sometimes it's confused with health informatics. So in bioinformatics, we don't deal with hospital information systems. So the hospital information systems, personal medical records, those data, it's not bioinformatics, okay? Or X-ray images, biopsy images. I mean, if here is really critical. There are some critical words that are underlined here. Biology in terms of molecules. So it's really bioinformatics mostly deals with life science problems at the molecular level. So the material, the domain is molecular biology and genetics. And we apply informatics or computer science techniques derived from disciplines such as math, statistics, computer science to understand and organize the information associated with these molecules on a large scale. So probably this on a large scale is the part that differentiates bioinformatics from computational biology. Computational biology sometimes create mathematical models of small biological systems. But when we have this data, large-scale data, and we try to interpret it, we try to cluster, get infer some new knowledge from this large-scale data, we usually classify this research domain under the title bioinformatics. Bioinformatics itself contains many subdisciplines, sub-areas. For example, as a computer science person, if you're working in the area of natural language processing, there are problems like text mining that you can apply to bioinformatics. Or if you're working in algorithms, string processing, you can work in bioinformatics. There are problems related to string processing algorithms in bioinformatics, large-scale strings. There are problems on computational structural biology, like trying to find three-dimensional shapes that are similar to each other, or trying to generate three-dimensional accessible surfaces of protein structures. Or if you are dealing in graph theory algorithms, there are also problems in bioinformatics. So there are many sub areas of bioinformatics, which is evident from the prominent journal in this area, the Bioinformatics Journal. So if you wonder about what people are doing, what research problems are there currently in bioinformatics, I suggest you go ahead and visit this journal website. Okay, and you can take a look at the latest issue. And our university has a subscription, as you can see. It recognizes my IP address, so Middle East Technical University Library. So some of the articles are not open access. Even non-open access articles we can access if we access this by using. If you're at home, you can use our university's VPN, virtual private network, which is the information I think is on Computer Center's website. So you can access these articles. So if you look at the journal contents, there are different subsections like genome analysis. That's a good question. Again, are there job opportunities for bioinformaticians except for the research? There are companies which they have, but still it's part of what they do is research and development most of the time. So bioinformatics is really like a research area. But in some of those companies, people just use existing bioinformatics tools that are published here. So sometimes bioinformaticians may find positions in companies, drug design companies or other companies that as a bioinformatician. But mostly there are research labs around the world, some of them government funded or some of them part of the universities. In terms of job opportunities, I'm not so sure how popular is this area, all right? So, but it's mostly research. Even if you're working in industry, it's mostly research-oriented things that you do. But sometimes you are a bioinformatics user, but sometimes you're a bioinformatics developer, developer of new algorithms. And if you look at different sub areas, there's genome analysis. The term genome may be new to computer engineers. It refers to the entire genomic sequence of an organism. So when we talk about whole genomes, we think about the entire chromosome material, the entire nucleotide sequence that form the organism. That's what genome is. It's this ome at the end. It means it's a collection of things, the entire repertoire of the genes. And you'll see this in other attached as a suffix in other terms like proteome. Proteome is the entire set of proteins in a certain state of a cell, or transcriptome, or interactome, all right? Metabolome. So this ome means the collection of things. So genome means it's the collection of genes. Yeah, you can annotate this. Okay, so sequence analysis. So let me. I want to get. All right. So how can you? I don't want to annotate right now. So how can I turn off? Yeah, right here, this one. But if you want to annotate stuff, you can request. Okay, sequence analysis. So individual genes or sequences. So genome, as I said, is the collection of genes. And there's phylogenetics. The second hour class that we had on Monday, it was for building phylogenetic trees. So phylogenetics, again, is about relationships of different genes, organisms with each other. There is structural bioinformatics. So look at these sections: structural bioinformatics, gene expression, or systems biology. Actually, these titles are really nicely also tells how this course is organized, starting with sequence analysis in the first weeks, and then also we're going to talk about phylogeny analysis, structural bioinformatics, and then gene expression analysis, and then systems biology. Systems biology has graphical data, relationships between these different entities. And as you can see, all these different areas require different computational tools, resources. And if you're an expert in one area, it doesn't mean you're an expert in the entire bioinformatics. You can conduct research in systems biology and have no idea whatsoever about genomic sequences. That's fine, all right. You may be a graph theoretician, and you may develop some graph theoretic algorithms that will help solve problems in systems biology, but you may have no idea about what protein structures are. So if you can take a look at these different articles if you're interested. And there's also data and text mining part of bioinformatics that I said, if you're if you're working in NLP, natural language processing, there are problems in text mining. Why is text mining a sub area of bioinformatics? Because there are articles, free form, lots of thousands of articles, scientific articles written in the area of molecular biology and genetics. And these articles that may be research reports of some low throughput, low scale, but highly accurate studies contains really very accurate and very valuable information. As the bioinformatics is trying to gather everything together in a data-centric manner, collect all this information, it will make sense to be able to get the information contained in these articles in an automated manner. So this is what data and text mining area is usually. How can we automatically capture structured data from free-form research articles, for example, by reading all those thousands of papers that are published in journals like Science, Nature? Can you find, for example, whether a certain protein interacts with another protein in certain settings? How can you get that information out of that material? You're not going to have text mining in our class, but that's, as you can see, a sub-problem. There's also image informatics. You asked on Monday's class whether there's a sub-area of bioinformatics related to images. Yeah, bioimage informatics is that area. And if you, for example, if you take a look at this, but the images are this complex filamentous network images. What is this? Let's see. Let's look at some figures in this paper and see whether they are images of large-scale organs or what are they? There, you'll see that in bioimage informatics, we are still at the molecular or single cell, at most maybe at the tissue level. But if you look at the PDF, actually, I don't need to look at the PDF. We can see it on this site. Yeah, let's see whether there are any figures here that will help us. Yeah, look at some figures are here. Some cellular images, maybe cell boundaries. Okay, so we are really at a very low molecular level, at the atomic resolution sometimes. So this is what bioimage informatics is about. But again, this is one part. This is an area that we will not focus in this class. In this introductory course, we'll not touch on bioimage informatics. All right, so Yeah, I think these are like, oh, actually, this is even, we may be looking at inside a single cell. For example, they may be microtubules. I'm not so sure. Do they? Let's micro. Yeah, cytoskeletal. So cytoskeletal means these are we're looking at the protein complexes that are within a single cell that makes up the skeleton of a cell, like these filaments, like microtubule, yeah, there's the microtubule networks. Okay, those are actually what you're seeing there are really atomic resolution, very small. I mean, you don't see the individual atoms, but we still these are very large complexes, protein structures that make up the skeleton of single cells. All right, so and it's a practical discipline with many applications. The problem domain is the problem domain is the molecular biology and genetics, and the problems come from there. And this is challenging because sometimes the problems can be solved by very simple computer science techniques, but sometimes you need to develop algorithms that may also contribute to computer science. You may need to develop new data structures. Some computational problems from this domain may be really interesting, and they may also increase the knowledge in computer science in general. But it's really difficult to find such problems which will contribute both to computer science and to molecular biology and genetics. So let's start with introductory biology for computer scientists who do not know what genes are. What is the data that we are going to be dealing with here? So this may be very simple. Just I'll try to do this in half an hour. So it will be a very huge obstruction. As computer scientists, we like to generalize abstract things. So it will be just a summary of data that we will be using this semester. One good thing, a very interesting thing that is shared by all life on Earth is that the DNA, which contains the information that make up these complex organisms, the DNA itself is discrete, which means that it's composed of nucleotides that can be considered like recipes, like instructions that will tell us how this organism is going to function. What it will be different to what will make the butterfly different from a fish. And really the information that is propagated from generation to generation really is this DNA. And this DNA really is a discrete information. And what happens is that, so DNA contains instructions to synthesize proteins in the cell. And protein, up to now, you may just heard the term protein as some nutrition information on what you eat. But in terms of nutrition, those proteins, food that contain protein, we use that protein actually for their building blocks, which are the amino acids. And in order for those proteins to be useful in our bodies, we need to make our own proteins. And how these proteins are the main functional things in a cell. A protein can act as an enzyme, as a channel on the cell membrane. Maybe in high school, you may remember the living things have a cell membrane. Animals, for example, we have a cell membrane. It's semi-permeable. Some things pass, some things do not. But we really didn't think about how certain things pass, how certain things do not pass. Is it about granularity? Or are they like things, if it's small enough, can it pass? It's actually not like that. The lipid membrane, there are certain proteins that are embedded inside the lipid membrane that allow passage of certain molecules, like certain ions. And so certain proteins, they act like ports. Some proteins are ports. Some proteins, we've just seen that cytoskeleton, they are used to build the skeleton of the cell. Many of them act like enzymes to catalyze reactions, chemical reactions that go on in the cell. Some of them are like switches that help to trigger things. Some of them are like signal transducers that are, there's a signal that is outside of the cell, and that signal has to be propagated inside the nucleus, for example. So by a cascade of interactions between different proteins, they propagate that information to the cell. So it's really what happens in a cell is really lots of proteins, thousands of proteins working in harmony together. And all of these working harmony, there's no intelligence actually. The intelligence comes from the physics and chemistry. So, but the theoretical physics and chemistry cannot explain what goes on in the cell because it's so complex. A protein like this you see here is composed of thousands of atoms. So that's probably one of the reasons the area of theoretical biology is not that advanced, because physics and chemistry help only to a certain regard. When you try to look at the big picture, the system gets so complex that even understanding what at the atomic level, understanding what goes on in a single cell is very difficult. So what we usually do is we try to perturb the system. As molecular biologists, one of the experiments that they do is, for example, what does this protein do? They can design experiments to knock down that protein. So what happens to the cell if we take it out? Okay, so that's one experiment that is conducted regularly to see, okay, if you take it out, oh, okay, it makes wings, so this protein is probably help you to make wings. But it's not that easy all the time. It's not a single protein that is responsible for the entire wings. It's, as I said, the whole entire blueprint in the cell is processes happen because of different entities act together within the process. So there are combinatoric pairwise interactions between them. So understanding life, what goes on in cell, is therefore really difficult. I mean, it's a very complex system, but the good thing is that we are getting every year with these advances, we're getting to the point where we understand most of the stuff what's going on in the cell. Okay, so the research in this area is getting really, really incredibly fast-paced research. We get incredible results with, for example, CRISPR-Cas9 technology. We can edit this information, not just knock down the protein, but we can say, what if I change this code? This is like the source code we have. It's like, yeah, this is what happened in the 2000s, actually. Since 2000s, after the whole genome of organisms have been found, it's like human society, I mean, as human beings, we have come upon the source code folder for life, all right? Now, this is really exciting, right? I mean, imagine you have all of your favorite games that you played up to now, and with some work, actually, these sequences, it's not by accident we have found these sequences, it's years of research. But imagine we have found the source code of all of your favorite games. So it will be really interesting to see how these games work. What are the parameters? What happens if you change certain parameters? How can you get yourself infinite lives in that game? So you try to analyze the source code. And this is exactly what we've been doing in the last 20 years. This instructions in the DNA, it's written in a language that we don't know. It's not C++, Python, but it is definitely a language, and it's discrete. So, for example, if you go ahead and look at certain databases like UCSC Genome Browser. So there's some data that is all of this. Most of the data is also publicly available. Like if you're interested in the source code for human, you can go to here. This portal. There are many portals like NCBI and NIH in the United States, EMBL in Europe. But look at this genome browser, interactively visualize genomic data. So this is the Google Map portal for our genome. And these are the mouse, rat, zebrafish, fruit fly, worm, yeast. So these that you see here on the right are model organisms, which means that certain things that we study on these organisms are easier to study. You don't feel bad if you kill a fruit fly, especially worms, or maybe you may feel bad about killing rats, but it's definitely better than killing humans, right? And in research, when you're doing research, when you do ask questions like what happens when we knock down this protein, it may kill the organism, and you don't want to do it on human beings. So that's why these are called model organisms, to find out what proteins do, what will happen, to understand. Again, our goal really is to understand what this entire source code is doing, and we're very far away actually still from achieving that, but we are also advanced a lot. We know, for example, the boundaries of functions in the source code. We know, okay, this is a function definition. Again, I'm making an analogy when I say function in terms of a C programming language. We see, okay, this must be a function that is written there, and it's called again and again. So it may mean something. So we can understand source code boundaries. We know what certain functions do, certain genes do. Each individual gene in an organism is like a function. And just think about the human genome, for example, contains about 25,000 genes. Just imagine you have found a source code of a game, which in the source code, there are 25,000 functions. How complex is that source code? Okay, yeah, I mean, the source code is that complex, so that's why it's so difficult to understand what's going on most of the time. So, but many of those functions are annotated, are known. For example, let's type in this chromosome position, chromosome 5. So this is like the latitude, longitude you have in it. Oh, it's already here on a map. So chromosome 5. 139 million, and this is like 40,000, about 40,000 bases, so 40,000 letters. So this is, oops, these are my previous, so let me just clear the tracks. So I use this UCS genome browser for, oops, so let's call disconnect. I use this UCS genome browser in my research regularly. So there are certain tracks that we do not. So this is chromosome 5 between this. Okay, so here this is a gene CXXC5, right? We on the same, yeah, chromosome 5. If you look at this, there's some visual information here that tells us about how this gene is organized here. If you zoom in enough in this gene, okay, let's do that again anyway. So we may end up in territory that is not meaningful. But if you look at this at the bottommost level, at the highest resolution, we have A, C, T, G, A, G, G, C, T, C. This is the genomic sequence. The coordinates are here from 105 to 110, 5 bases here between these intervals. So the length of like 15 centimeters you see on your screen corresponds to these 5 bases. So you can zoom in, zoom out, and the human genome, the length of the human genome, the entire repertoire of the collection of all the chromosomes is about 3 billion. So 3 billion characters organized like this. And this source code you see is called the reference assembly. And human beings have differences in these regions. And another interesting thing is, in this gene, for example, what happens if this G is changed into a T or an A? Will it change? So you're changing different parts of the source code, and some of them make you susceptible to certain diseases. One important research in this area in bioinformatics is when you get source codes of many individuals, like 10,000 individuals, you get the source code. Where are their similarities? Where are their differences? And in those differences, can we associate those differences statistically significantly with certain diseases? For that, you design experiments where you collect individuals, maybe 5,000 of individuals do not have that particular disease. The other 5,000 have that particular disease. So what are their differences in terms of this genetic code that we can pinpoint maybe causing this disease? So this is really, the data is huge. We have the whole genome sequences of thousands of organisms available, and some of them we know their protein sequences. So DNA is ACGT. 3 of those ACGT letters in a frame, they code for amino acids, and there are 20. The language of protein sequences is 20 letters, 20 amino acids, and those amino acids in the cell, actually, they form a 3-dimensional shape like you see here. And that 3-dimensional shape, the amino acids that are at the surface, their physiochemical properties, whether they are charged, polar, non-polar, it really affects lots of things. And the shape of the protein really matters. And knowing protein structure shapes, for example, is another thing that is very important. And one database for that is RCSP. So if you go to RCSP, so this is the March molecule of the month, cisplatin and DNA. So one protein here, the blue one, I think is the protein, is interacting with the DNA. So there are certain proteins which are made up of amino acids that physically interact with the DNA to initiate or block certain processes. So, and these are all at the atomic level. Each of these circles that you see here, spheres that you see here, probably correspond to an individual atom. So we are at this very low level. Look at this. This is the large version. And if you look at these visualizations, they don't look like photographs to me. Who drew this? Okay, they're like cartoons, hand-drawn by someone. So this is really what happens. You cannot have a photograph of this because they're at the atomic level. Some of the things that you see here have size less than the wavelength of visible light. So so we cannot really visualize these atoms. They cannot be visible, but they have force fields around them to make bonds with different atoms and form these macromolecular structures. So if you click on the PDB link, for example, of one of these, it will give us a different view. If you look at this, I can even probably view the structure interactively. It's 3D structure here. Okay, look at this. So this is a protein structure. It just looks like ribbons and lots of coils here. And I think this is the DNA that we are currently interacting. But as you can see, it's a 3-dimensional shape. Now, this representation, the cartoon representation that we see here, is to help biologists to understand different parts of this 3-dimensional shape. Actually, this is not how it really is inside a cell. It's, as I said, it's made up of small atoms that have force fields, and probably there are options here to change the view. All right, so this is the structure. I'm not very familiar with this interface. There are component structural motif search density, so there are different things here. There should be options here to view the structure in different visualization options. Probably it's below here. Select the different, maybe JSML. Well, let's see. Oh yeah, here, it's here. It says cartoon model, okay? Now, I probably can change this cartoon model to a different representation right here. The type cartoon. Can I change this? Yeah, let's change this to ball and stick. Look at, yeah, balls and sticks. So what you see here are now the view is changed. This is the same protein structure, but now viewed entirely differently. So these are just visualization styles that help biologists in different tasks. Gaussian volume or space fill. Yeah, and these are the individual atoms now we see. Look at them, our atoms. And coloring, I can have it by atom property, element symbol. Now every element has a different color. Okay, I changed the coloring here. We don't see the individual amino acids, but we see the atoms. There are carbon atoms, nitrogen atoms, oxygen. So oxygen, carbon, nitrogen, those are the main atoms mostly you see, and hydrogen. This is an organic molecule, as you can see, and this is a protein basically, and it contains and this is probably small, some larger ones contains thousands of atoms. All right, so we will talk about these protein structures when we come to that week about protein structures. Just to show you this, what makes this protein structure, the information is contained within the DNA. It makes a certain type of protein, which is specific to that organism most of the time, and those repertoire of proteins most of the time makes these different phenotypes, the observed properties of these organisms. And even the organs, they differentiate. One very remarkable thing in the source code is that our different organs, different tissues within the same organism have different requirements in terms of function. So the single source code installed in all of the cells in our bodies, it's run differently. Consider this, the whole genome as an operating system. An operating system knows which parts to execute depending on its environment, depending on which tissue it's in. We have the same source code in our neuron cells, same source code in our muscle cells, but neuron cells and muscle cells are really different in terms of the proteins that are required or executed. So this is called sometimes gene regulation, mean that the cell knows how to regulate itself, which functions are active, which functions are not active. So this is a very simple picture of the animal cell. I'm going to skip most of the slides here. And the data, that instruction, the genomic data looks like this. And imagine this is a really long string of 3 billion letters for the entire human genome. It's really difficult to analyze and make sense of it. So this is the first initial source code that we got in 2000s. And then by developing certain algorithms, we identified certain functions that start and end different places, which are genes. A single gene is responsible to encode a protein. And sometimes different variations of that gene can be used to encode different versions of proteins. Okay, so it's not always one-to-one, but really we can consider this as the instruction set to encode the proteins. So here, as I said, proteins have 20-letter alphabet, 20 different amino acids. These are single-letter amino acid codes. There are also 3-letter amino acid codes that, for example, this is leucine. It's 3-letter code, maybe LEU. So the amino acids, they are actually molecules that contains like 20 different number of atoms. Some of them are small, some of them are larger than the others. But a chain of amino acids is used to make up those proteins. Proteins, their shapes are important. We are going to talk more on protein structure again when we come to that week. But just know that this is a different type of data. One of them is just linear sequence, one-dimensional information. On the other hand, we have three-dimensional information when we talk about proteins. We're going to come and talk about these things later. And yeah, these are the three-letter codes that I mentioned for leucine, for example. These are the different amino acids. And as you can see, the three-letter DNA coding to an individual amino acid is there are some redundancy here, which means that our certain parts of our genome are immune to like change. For example, if whether it's CGC or CGA or CGT, if in that frame this third nucleotide is changed to something else, still it will be read as arginine, and in the final product protein, we will see no change. Certain mutations will not affect at all at how we function. And I also, the array data, I mentioned this a little bit on Monday, last Monday. Another data that we have is all these functions. So we have the source code. This is like static analysis. If you know software engineering, analyzing a source code as it is at the source code is like analyzing the genome. Now, runtime analysis is something like you let the program run, and you look at an operating system, and at a certain state of the operating system, you ask this question: which functions are currently active? Which function is calling which function? And that data is also available to us as bioinformaticians because there are biotechnological experiments or devices that can gather that data for us at this specific instant, which genes are expressed into messenger RNAs. And in a sense, when we have the messenger RNA, it means that it's translated into a protein. So we can query these things on a large scale, and that's gene expression data sets. We're going to, again, talk about the details of these when we come to the corresponding week within the semester. So I'm skipping these parts there is phylogeny, there is organizing. Not everything within an organism is related. Organisms are also similar to each other. We have similarities with all those model organisms. Some of them are single cellular. All right. So, and the data really is huge. And we are going, in this course, you'll have a chance to experience with some of these data. All right, I think that's all I want to mention. You can read the remaining of these slides on your own. Let me know if you have any questions. And computer science is really critical to biology because since we don't have this theoretical biological background, all this data, all this complex system, computer scientific algorithms, techniques are used, models, algorithms are used to make sense of what's going on in biology. All right, now there is the scalability challenge, lots of data. As I said, I'm going to skip these slides, and this is my last slide. There are different problems. There are challenges, like the DREAM challenge that asks different researchers to solve different molecular biology problems. You may have heard this deep fold that we mentioned last time. It was the protein structure prediction challenge. If I give you the sequence of amino acids that make up a protein, can you predict what three-dimensional shape it's going to have? This was a really difficult problem, but deep learning also helped solve this problem. Yeah, and starting next Monday, we will start with our first problem. Given two genetic sequences, DNA sequences or protein sequences, maybe from two different organisms, which we know they're functionally related, can we align them on top of each other so that we find the conserved similar parts and differentiated parts on the sequence with efficient algorithms? So we will start with this problem on Tuesday. And if you need more introduction to molecular biology and genetics, I suggest On the course website, there are lots of reading material for the first week. There's introduction to, but this one, Bioinformatics and Introduction for Computer Scientists, is a really nice article that is geared to, that is written for computer scientists. I so entered wrong, 5710465 underscore. Yeah. So you may read these articles. Okay. It will be a really nice introduction to molecular biology and genetics. And there are also chapters from our textbook, how cells and genomes, how they read. You don't need all the details, but just know when we talk about what a gene is, we are referring to that instruction at the DNA material that we are referring to. When we talk about a protein, it may have an amino acid sequence. It also have a 3D structure that has certain functions within the cell. So these are the main terms that we will be using throughout the semester. If you look at the entire weekly schedule, just knowing the terminology and what is what will be sufficient in this regard. It's really, I mean, all of these material that I just tried to summarize in a couple of, in a half an hour, there are whole semester courses in the molecular biology and genetics department. If you're interested in those, you can also take classes from that department if you want to learn more details about these things. But our textbook contains also a lot of nice material. Okay, so I'm going to stop recording now.